Hi, my name is Shannon, and I am a master's student in the Hlusky Lab in Northern Arizona University. And today I'll be talking about my project, which is looking at browsing effects on the rare species Salix arizonica, or Arizona willow. So first off, to set up the story, it is to say that it is a story of economics. And that is, with growth trade-off theory, plants have a limited number of resources that they can use. So primarily they like to use these resources to allocate to growth. However, when they're under attack from herbivory or pathogens, then they induce defense compounds. And when this happens, they take away those resources that they would use for growth, and therefore they're more so stunted. We wanted to look at this theory with Salix arizonica because it is a species that has been of critical concern since the early 1990s. It is found only in the Four Corner States and high elevation wetland meadows, and it is listed as impaired in Arizona and Utah, or critically impaired in New Mexico and Colorado, which essentially this means that it's at risk of extinction due to limited population sizes and small habitat needs, and other external threats such as recreation use, change in hydrology, climate change, and what we're going to be looking at for this study is browsing effects by ungulates. So here's a map of the distribution of where Salix arizonica occurs in these little dots. You can see that they are pretty far apart from one another. And so I have my questions titled here, and they're also labeled on the top of the slides for your reference for the rest of the presentation. So for my first question, I wanted to see whether defense compounds are reduced when individuals are protected for a long period of time. And then for my second question, was more of an experiment, was to set up short-term protection for one season of browsing to occur and see if there's a plastic response in reducing defense compound production. And then lastly, I wanted to see whether there's a growth trade-off with defense compound production, and I'll be using height for this approximation. So when I was out in the field in both seasons, I collected leaf tissue samples and I took down notes on various things such as phenology, and took down browsing severity categories. So with my top illustration, it shows whether an individual is browsed at all or up to heavy, which is more than 50% of the total stems being munched on or not. And then because of preliminary genetic research, there is evidence that each of these populations, which are outlined in the large colored polygons, are genetically dissimilar from each other. So they're genetically unique from one another. So I'll be using this as an indicator when I'm looking at the growth trade-off effect. And then the smaller black circles are what I'm calling my sites. And here in Arizona, the White Mountains, I set up a game cam to capture evidence of current activity of browsing. So at day, we see that there's cows browsing, and then at nighttime, that's when the wild ungulates seem to come browse these systems. So after I collected my samples, I extracted the compounds with methanol, ran my lab analysis, and saw that there are three main compounds in Salix arizonica, which are called phenolic glycosides. I have salicin, salicordin, and HCH salicordin, and plus there's two unknown compounds as well. And for my analyses, I just took the total sum of each compound for what I'll be using either as a predictor or a response. So to start off with my first question, to look at whether there is an effect in reducing the defense compound or phenolic glycoside concentration with long-term protection, I'll be only using the Arizona samples because it had the largest number of protected individuals with a good sample of unprotected individuals as well, as you saw in the previous game cam slide. And here is also what you would see out in the field in the White Mountains is these individuals that have been protected for a long time on the left-hand side. They are outgrowing their exclosures and they're still being browsed, or ones that have been exposed to browsing over decades, we see that the graminoids are overcrowding them and they typically are around, I don't know, 6 inches to a foot tall. So this is like the variation that we see out in the field. And after running my analyses, I saw that regardless of browsing occurring on caged individuals, there is a significant effect of reducing the phenolic glycoside concentration, or the defense compound concentration. So with my right-hand figure and with the rest of my figures for this presentation, each dot is indicating that it is an individual. And the total confidence interval for the caged individuals is lower than the uncaged individuals. And then for my second question was whether new protection allowed for plants to reduce investment in defense compounds. So for this, I primarily focused on the New Mexico population, the yellow polygon on the right-hand side, and each star indicates a site where I set up new exclosures or reinforced new exclosures. But due to wildfires and other issues, I also had a site in Utah as a backup in case I couldn't fulfill my target question. And I wanted to focus on New Mexico because it seems that genetically there's less hybridization there compared to Utah and Arizona. And here is a smattering of what it looked like when we're installing new caging infrastructure. So either I protected individuals individually with something called a chicken wire cloak, or I installed some T-post horse fence, or as well tried to reinforce existing exclosures that were just dilapidated. And sometimes it didn't work. Unfortunately, ungulates are really strong and they're able to break through things, or even like whether possibly broke stuff down. I am not very sure, but I imagine it's a combination of both. So with that, some of my protected individuals ended up getting munched on. So for this, then I had to be creative and look for individuals that didn't have any evidence of browsing. So I looked only for current year browsing, and I did this by looking for the young green shoots and whether they had evidence of the characteristic of ungulate browsing or not, which we can see in both of these pictures. And then after running my analyses, we saw in both populations that there was not a reduction in defense compound production, whether they were caged or uncaged. And then we saw this across each of the compounds as well. And to just reiterate, the top blue points are the Utah samples and the bottom ones are a combination of my New Mexico sites. So then for my last question, I wanted to look into whether there was a growth trade-off between willow overall growth versus the production of defense compounds. And again, I'm using height as an indicator for this. And to reiterate, the growth defense trade-off theory is that plants have a limited number of resources, so they can either use most of those for growth, some of them for defense, or most of those for defense, and then therefore they might limit their growth production. So first I wanted to illustrate that if you look at it by population and you look at browsing severity against the production of defense chemicals, you see that even with increasing browse severity, there isn't a significant increase in phenolic glycoside concentration. However, if you look at plant height against browse severity, we do see a significant reduction at the highest browse severity. So the way I wanted to look at this was to use the defense compounds or the phenolic glycosides within the principal component analysis, which is a multivariate analysis that just combines everything and reduces down to like a few different dimensions that make it easier to interpret. So here we have the first two dimensions on the x-axis and the y-axis, and this shows about almost 70% of the variation that these chemicals are expressing and interacting. And I think the interesting thing here is that if you break it down by population, or here I have it as state, they still cluster with each other, which is really cool. So what I wanted to do then was then take each of those populations, which you see here again on this map, and rerun analyses with a PCA per population. So first off, looking at New Mexico, if I plot the total phenolic glycoside concentration with the principal component analysis number 1, which explains the most amount of variation, you see that all of these dots are actually each of the individuals that I've collected, and that there is a very strong correlation between the PG concentration and the axis. So we see that on the left-hand side, when the axis number increases, so does the PG concentration. And so then when we plot this with height, we see that the PC axis is highest when the plants are shortest. So there is an illustration here that there is a growth trade-off happening. We see the similar relationship as well with our Arizona samples as well. However, even though we see that there is this increasing defense compound concentration with the increasing in the PG or PC axes, we don't see this relationship with the Utah samples. And also on this line, I'm indicating the maximum PG concentration for the New Mexico and Arizona populations, but Utah has a threshold that goes far higher in these defense compound concentrations, which coincides with the theory that with hybridization, plants can possibly increase their phenolic glycoside concentrations. And a different way I wanted to look at this was to run a model with everything together and take into consideration browse severity, the population, and the KH effect. And once you run this all into the model, you see that the only two things that significantly affect plant height is KH and PG production. So when I graph this as well, you can see that on my x-axis with the phenolic glycoside concentration, the lower it is, the taller the plants are, and the higher it is, the shorter the plants are. So to recap, defense compounds are significantly reduced when they do have long-term protection. However, we don't see this effect in the short term. And as well, there is in fact a growth trade-off with an increase in defense compound concentration. And what I gather from this is that these plants need a longer period of recovery time before we see these high grazing introductions into their systems. And having this over and over again, plants aren't able to recuperate easily. However, even though this is like a conclusion I'm trying to draw right now, there definitely does need to be more experimentation in order to confirm these ideas. So with that, I wanted to thank you guys for listening and for helping fund this research, along with New Mexico Native Plant Society for helping with funding, USGS and NAU, and everyone who's been support. So again, thank you.